Today I am taking up the topic genetic disorders. This topic is being held under two headings or two parts. Part 1 deals with genetic disorders and part 2 deals with problems associated with genetic disorders. Today let us take up part 1, genetic disorders. The genetic disorders in human beings are two types: Mendelian disorders and chromosomal disorders. Mendelian disorders are due to a single mutation occurring either in the somatic chromosomes or in the sex chromosomes. But the chromosomal disorders are due to a change in the number of chromosomes. First, let us take up Mendelian disorders. The Mendelian disorders are so called because they occur in the pattern of Mendelian inheritance. It means they follow the laws of Mendel. Mendel conducted his experiments on the pea plant. Then, the Mendelian disorders occurring in human beings cannot be studied like Mendel, because in human beings we can't carry out some classes like back cross, test cross, or we can't inbreed. Then the sample size is very small and the time taken for one generation is very long, 20 to 30 years. So in human beings we can't conduct experiments just like Mendel conducted on pea plant. Then how to study or how to understand the Mendelian disorders in human beings? There is a method for this, that is analyzing the pedigree trees or family trees, or we call that pedigree analysis. So sometimes the family history is recorded for over many generations. Study of such family history is known as pedigree analysis. In the family trees, they represent who married whom, how many children they have got, how many children suffer from diseases, like that. They include all the details of a family over many generations. Then, in the pedigree analysis, some standard symbols are used. Here, a square represents a male, a circle represents a female, a diamond shape represents sex unspecified. That means, if the fetus is aborted, the sex is not known, then this symbol is used, diamond symbol is used. Shaded symbols indicate affected individuals. That means they are affected by some heritable genetic disorder. When the male and female are joined by single line, it is called mating or marriage between two persons. If a male and a female are joined by two lines, it indicates it is a marriage between two closely related persons, or a cousin marriage. Okay, cousin marriage. Then here, on the top line, we represent the male and the female, and the second line represents the children. In this case, the shaded square represents the affected male child. Like that, the family trees are drawn. Now let us come to Mendelian disorders. I have already told you, Mendelian disorders are due to a mutation occurring in a single gene. The mutation may occur in a somatic chromosome or sex chromosome, and the mutation may be dominant or recessive. Then, how to understand them? 3 examples are given in the book: haemophilia, sickle cell anemia, and phenylketonuria. There is another one I have written here: color blindness. Color blindness is just like haemophilia. The pattern of inheritance of color blindness is similar to haemophilia. First, let us take up haemophilia. Haemophilia is a sex-linked recessive trait. This disease is transmitted through a recessive gene present in the X chromosome. So it is known as sex-linked recessive trait. In human beings, you know that females have two X chromosomes and males have one X chromosome and one Y chromosome. Females always transmit one X chromosome to the son and one X chromosome to the daughter. Coming to the males, males always transmit their X chromosome to their daughters and Y chromosome to their sons. It means mothers transmit their X chromosomes equally among daughters and sons, whereas the father transmits the X chromosome always to the daughter and Y chromosome always to the son. This point you have to remember very carefully. Then, what is haemophilia? Haemophilia means blood fails to clot. One of the factors involved in blood clotting is absent or has undergone mutation. As a result, blood fails to clot. Normally, when we are injured, within 5 minutes, blood clotting takes place and hemorrhage is stopped. But in hemophilics, even a little injury or a small cut causes them to bleed to death. They lose blood continuously and they die. For the first time, this disease was discovered in the family of Queen Victoria. So it is known as royal disease. It is also known as royal disease or bleeder's disease. Bleeder means who bleed continuously to death. Then, to become a hemophilic female, that woman must receive both the recessive genes. It is possible only when her mother is a carrier, because hemophilic mother is rare, her mother should be a carrier and father should be hemophilic. So only when a mother is a carrier and father is hemophilic, such women become hemophilic. Then, hemophilic women are very rare because even though they survive, at the time of first menstruation, they bleed to death. Hemophilic men are usually found or somewhat common. They survive because they don't have any problems of these menses. Then, a hemophilic mother, a carrier mother, carrier for hemophilic disease, can transmit her mutant gene either to a daughter or to a son. If the son inherits the mutant gene, he exhibits hemophilia. If the daughter inherits the mutant gene from mother and a normal gene from father, she will be a carrier or she will be in a heterozygous state and she cannot or she will not exhibit the hemophilic disorder, but she is a carrier. That point you have to remember. Then, the second one is sickle cell anemia. It is an autosomal recessive disorder. It is transmitted by a recessive gene present in the autosomes. So it is known as autosomal recessive disorder. Here, both the parents contribute equally for the disease. Then, in our case, RBC are disc-shaped and they transport oxygen to the tissues. Sometimes what happens is, a mutation occurs in the one of the gene responsible for the synthesis of hemoglobin. In the hemoglobin, there are two polypeptide chains, alpha chain and beta chain. The mutation occurs in the beta chain. In the beta chain, in the 6th place, there is one amino acid known as glutamic acid. Its triplet code is GAG. So normally, GAG triplet code is present in the 6th place of beta chain and it is responsible for the incorporation of glutamic acid. But a mutation occurs here and this mutation is called point mutation. In the case of point mutations, a change occurs in a single base or a single nucleotide. Here what happens, Instead of A, it is replaced by U. That means A means alanine is replaced by uracil. As a result, instead of glutamic acid, the amino acid valine is incorporated. So in the case of beta chain, in the sixth place, because of the point mutation, valine replaces glutamic acid. And it is a serious issue. What happens? In heterozygous state, there is no problem. The persons appear normal. But in homozygous state, what happens? Under conditions of hypoxia, that means under low oxygen tension, the RBC, they lose their disc shape and acquire sickle shape. If the RBC are in a disc shape, they can travel easily through blood capillaries. But once the shape changes to sickle shape, they clog. They can't pass through the capillaries. They clog and cause complications. This problem usually arises while climbing mountains or when the people reach higher altitudes where the oxygen tension is low. So this disease appears only under hypoxia conditions, under low oxygen tension. Then there is one advantage also here. People who are heterozygous for this disease do not suffer from malaria, especially from falciparum malaria. That is one advantage of sickle cell anemia. Here I have used two terms HbA and HbS. HbA means adult hemoglobin. HbS means sickle cell hemoglobin. So due to this mutation, point mutation, HbA becomes HbS. That means adult hemoglobin is changed into sickle cell hemoglobin. The third one is phenylketonuria. Phenylalanine is generally converted into tyrosine in the presence of the enzyme phenylalanine hydroxylase or PAH. So in the presence of the enzyme PAH, phenylalanine is converted into tyrosine. Sometimes what happens, due to a mutation, this enzyme is not synthesized. As a result, a metabolic block is formed. A metabolic block. That means this is not converted into this one. That's what is known as metabolic disorder. Metabolism involves a number of steps. Even when one step is affected, it results in the failure of that step, that synthesis of the end product. That's what is known as metabolic disorder. Here the metabolic disorder is due to the absence of this enzyme PAH. Then what happens? Then phenylalanine accumulates in the tissues and is converted into phenylpyruvic acid and other derivatives. And the formation of phenylpyruvic acid, what it does? It causes mental retardation. So in the long run, what happens? The persons suffering from the deficiency of this enzyme suffer from mental retardation. Their urine appears black in color, and their IQ, intelligent quotient is also very low. So all these symptoms appear and they are together known as phenylketonuria. These are about the Mendelian disorders. Coming to the chromosomal disorders. Here, chromosomes are either added, deleted, or multiplied in number. The cause is due to separation of chromosomes into daughter cells during cell division. During cell division, some abnormality happens and all the chromosomes do not enter the required cell, daughter cell. Here we have to remember some terms. Aneuploidy, loss or gain of one or more chromosomes is known as aneuploidy. In aneuploidy, one chromosome is added or deleted, or more chromosomes are added or deleted. Then polyploidy. Polyploidy means addition of a whole set of chromosomes. Addition of a whole set of chromosomes. We have two sets of chromosomes. Each set consists of 23 chromosomes. In polyploidy, instead of 2, we may have 3 sets or 4 sets like that. Polyploidy is not that common in animals, but it is very common in plants. Most of the plants are polyploid. Examples: sugarcane, maize, wheat, banana, apple, etc. Why polyploid occurs? Polyploid occurs due to the absence of cytokinesis after karyokinesis. So during karyokinesis, chromosomes are multiplied, and during cytokinesis, cell is divided. If cytokinesis is absent, what happens? All the chromosomes remain in the same cell, and this ultimately leads into polyploidy. Then there are 3 more terms you have to remember. Trisomy. Usually we have chromosomes in the form of sets. Set means homologous pair or homologous set. So usually homologous chromosomes occurs in pairs, but sometimes instead of 2, 3 chromosomes occurs in that set. That is known as a trisomy. So trisomy means instead of 2, 3 chromosomes occur in a homologous set. Monosomy. If one of the homologous chromosome is missing or lost, it is called monosomy. If both the homologous chromosomes are lost, it is called nullisomy. So aneuploidy means gain or loss of one or more chromosomes. Polyploidy means addition of complete sets of chromosomes. Trisomy means the same chromosome occurs thrice instead of twice. Monosomy means loss of one homologous chromosome. Nullisomy means loss of both the homologous chromosomes. Here 3 examples are given: Down syndrome, klinefelter syndrome, and Turner syndrome. These are named after their discoverers. Down syndrome, it is known as trisomy 21. That means the 21st chromosome occurs 3 times instead of twice. So here the karyotype is 47. Karyotype means chromosomal number. Our karyotype is 46. But here, because of one additional chromosome in the 21st set, the karyotype is 47. So in these people, Down syndrome people have 3 21st chromosomes. Then these affected persons appear normal, but they exhibit the following conditions. They are short in stature, they are mentally retarded, tongue is furrowed, furrows are present on the tongue, mouth is partially open, palm is broad, with a characteristic simian crease. In the palm we have some lines, but they have got a big crease running all along the width of the palm. Simian crease means it is present in the monkeys. Then the karyotype is 47. So these are some of the disorders exhibited by Down syndrome people. Second one, klinefelter syndrome. It is also trisomy, but trisomy of 6 chromosomes. These are supposed to be males. They possess XY chromosomes. Instead of XY, they got one additional X chromosome. That is known as trisomy XXY. The additional X chromosome causes so many complications. Because of an additional X chromosome, their karyotype is 47. They have overall masculine development, male development. They are abnormally tall. But they also exhibit some of the feminine traits like development of mammary glands. It's called the gynecomastism. So they develop female traits like gynecomastism. Their facial hair is poor. They have very little hair on the face. That is, moustache and beard have few hair. Then, these people are sterile. They fail to produce sperms, so they are sterile. Testes are also small. and why klinefelter people are produced. This is because of non-distinction during meiotic division. During meiotic division, what happens? The homologous chromosomes, after exchanging their segments, get separated. This is called disjunction. If they do not separate, it is called non-distinction. So the homologous chromosomes, the pairing chromosomes, they travel together. And this is known as non-distinction, and non-distinction results in klinefelter syndrome. The third one is Turner syndrome. Here, there is only one sex chromosome, that is X. Other one is absent. These are supposed to be the females, but one of the X chromosome is missing. Here, the karyotype is 45. Instead of 46, only 45 chromosomes are present. These are sterile females. Their ovaries are absent, and they fail to produce ova. They are also short in stature. The secondary sexual traits fail to appear. That means female secondary sexual characters do not appear in these persons. They have a webbed neck, and these persons are sterile. So that is about Turner's syndrome. In this way, various chromosomal disorders occurs in human beings. Thank you.